Hello everyone and welcome to this lecture on antibody-based methods for virus detection. Today, we'll explore two powerful techniques, ELISA and Western blot. Enzyme-linked immunosorbent assay, ELISA, is a powerful method for detecting and quantifying a specific protein in a complex mixture. ELISA is a widely used test for HIV detection. For HIV detection, blood, oral fluid or urine samples can be used. While blood and oral fluid ELISA tests are highly accurate, urine ELISA tests for HIV are less accurate. Confirmatory test is needed for positive results. Positive ELISA requires a Western blot test for confirmed diagnosis. Inaccurate results are extremely rare. However, it is possible in case of early infection when antibodies do not form or due to laboratory errors. This test gives high accuracy with combined testing. That is, ELISA and Western blot put together give a 99.9% accuracy in HIV detection. Now let us look into the requirements and procedure for performing an ELISA test. ELISAs are performed in polystyrene plates, typically 96 well plates coated to bind protein strongly. Depending on the ELISA type, testing requires a primary and or secondary detection antibody, analyte, antigen, coating antibody or antigen, buffer, wash and substrate, a chromogen. There are four main general steps to completing an ELISA immunoassay. These steps are coating with either the antigen or antibody. Second, blocking, typically with the addition of bovine serum albumin to block the unbound sites to prevent non-specific binding. Third, detection. The primary detection antibody is a specific antibody that binds to the protein of interest. In contrast, a secondary detection antibody is a secondary enzyme conjugated antibody that binds to the primary antibody that is not enzyme conjugated. Further, these antibodies are linked to substrates that can generate a color. There are many substrates available for use in ELISA detection. However, the substrates most commonly used are horseradish peroxidase (HRP) and alkaline phosphatase (AP). The substrate for horseradish peroxidase is hydrogen peroxide, resulting in a blue color change. Alkaline phosphatase measures the yellow color of nitrophenol after room temperature incubation periods of 15 to 30 minutes and usually uses para-nitrophenyl phosphate (PNPP) as its substrate. Between each of the four steps is a wash of the plate using a buffer such as a phosphate buffer saline and a non-ionic detergent to remove unbound material. The wells are washed 2 or more times during each step depending on the specific protocol. In a usual ELISA protocol, a serial dilution of concentrations of antigen or antibody is placed in the wells of the plate. After the results are measured, a standard curve from the serial dilutions data is plotted with the concentration on the x-axis using a log scale and absorbance on the y-axis using a linear scale. This is the final read. Now, let us look into the different types of ELISA. First, direct ELISA. Initially, in a direct ELISA test, which is considered to be the simplest type of ELISA, the antigen is adsorbed onto a plastic plate. Then, an excess of another protein, normally bovine serum albumin, is added to block all other binding sites. While an enzyme is linked to the antibody in a separate reaction, the enzyme antibody complex is applied to adsorb to the antigen. After excess enzyme antibody complex is washed off, the enzyme antibody bound to the antigen is left. By adding in the enzyme substrate, the enzyme is detected, illustrating the signal of the antigen. In an indirect ELISA, we quantify antigen-specific antibody rather than the antigen. We can use indirect ELISA to detect antibodies against HIV. The indirect ELISA starts with attaching the known antigen to the bottom of the microtiter plate wells. After blocking the unbound sites on the plate, the patient's serum is added. If antibodies are present, that is the primary antibody, they will bind to the antigen. After washing away any unbound proteins, the secondary antibody conjugated to the enzyme which is directed against the primary antibody, that is anti-human immunoglobulin, is added. The secondary antibody allows us to quantify how much antigen specific antibody is present in patient serum by the intensity of the color produced by the conjugated enzyme chromogen reaction. However, this may lead to non-specific signals because of the cross reaction that the secondary antibody may bring about. In a sandwich ELISA, the goal is to use antibodies to precisely quantify the specific antigen present in a solution, such as the antigen from a virus. The first step of a sandwich ELISA is to add primary antibody to the wells of the microtiter plate. The antibody sticks to the plastic by hydrophobic interactions. After an appropriate incubation time, any unbound antibody is washed away. Washes are used in between each of the subsequent steps to ensure that only specifically bound molecules remain attached to the plate. A blocking protein is then added. Example, bovine serum albumin or milk protein casein, to bind the remaining non-specific protein binding sites in the well. Some of the wells receive known amounts of antigen to allow the construction of a standard curve, and unknown antigens are added to the other wells. The primary antibody captures the antigen, and following a wash, the secondary antibody is added, which is a polyclonal antibody that is conjugated to the enzyme. After a final wash, the colorless substrate chromogen is added and the enzyme converts it to a colored end product. The color intensity of the sample caused by the end product is measured with a spectrophotometer. The amount of color produced is measured as absorbance is directly proportional to the amount of enzyme, which in turn is directly proportional to the captured antigens. These ELISAs are extremely sensitive, allowing antigen to be quantified in nanogram, that is 10 to 9 nanogram per ml range. Competitive ELISA is also known as inhibition ELISA or competitive immunoassay. Competitive ELISA is a technique used to estimate the antibodies present in a specimen such as serum. The principle of the test is that two specific antibodies, one conjugated with the enzyme and other present in the test serum. If the serum is positive for antibodies are used. Competition occurs between two antibodies for the same antigen. The appearance of color indicates a negative test, that is absence of antibodies, while the absence of color indicates a positive test, that is the presence of antibodies. The central event of competitive ELISA is a competitive binding process executed by the original antigen, sample antigen and added antigen. The procedures of competitive ELISA are different in some respects compared with other forms of ELISA. Procedure of competitive ELISA. In this test, the microtiter wells are coated with the antigen. The sera to be tested are added to these wells and incubated at 37 degrees and then washed. If antibodies are present in the test serum, the antigen-antibody reaction occurs. The antigen-antibody reaction is detected by adding enzyme-labeled specific antibodies. In a positive test, no antigen is left for these antibodies to act. Hence, the antibodies remain free and are washed away during the process of washing. When substrate is added, no enzyme is available to act on it. Therefore, positive result is indicated by the absence of color reaction. In a negative test, in which no antibodies are present in the serum, the antigen in the coated wells is available to combine with the enzyme conjugated antibodies, and the enzyme acts on the substrate to produce color. Advantages of ELISA: high sensitivity and specificity. It is common for ELISAs to detect antigens at the picogram level in a very specific manner due to the use of antibodies. High throughput. Commercial ELISA kits are normally available in a 96 well plate format, but the assay can be easily adapted to 384 well plates. Easy to perform. Protocols are easy to follow and involve little hands-on time. Quantitative. It can determine the concentration of antigen in a sample. Possibility to test various sample types: serum, plasma, cellular, and tissue extracts, urine, and saliva, among others, can be used as test samples. Disadvantages of ELISA: temporary readouts. Detection is based on enzyme substrate reactions, and therefore readout must be obtained in a short span of time. Limited antigen information. Information is limited to the amount or presence of antigen in the sample. Western blot. A Western blot, sometimes called a protein immunoblot, is an antibody-based technique used to detect the presence, size, and abundance of specific proteins within a sample. The technique was developed in 1979 by Harry Tobin and his colleagues, and later named Western blot due to the technique's similarity to Southern blotting used for DNA. Western blotting is a commonly used method for detection of HIV. It can be used for qualitative and semi-quantitative protein analysis. Western blotting has 6 steps, including sample preparation, gel electrophoresis, proteins transfer, blocking, antibody incubation, and proteins detection and visualization. First, sample preparation. Proteins can be extracted from different samples such as tissues or cells. Since tissue samples display a high degree of structure, the tissues are first broken down by mechanical disruption such as homogenizer or sonication. Protease and phosphatase inhibitors are commonly used to prevent the digestion of sample at cold temperatures. The proteins in the sample must then be separated. This is typically achieved by protein electrophoresis, such as sodium dodecyl sulfate polyacrylamide gel electrophoresis SDS page or native page, which separates proteins based on their molecular weight or charge. The specific separation method chosen will depend on the aim of the analysis. For a clean image, the samples are centrifuged to remove any solids in order to load only the soluble fraction. If the protein of interest is an insoluble fraction, example, cell membrane bound proteins, investigate pre-treatment methods to liberate and solubilize it first. Solids will impair the running of the gel, and it is likely that the protein of interest will remain in the stacking gel. It is also important to load appropriate control samples and size marker ladders to enable interpretation of the final blot. It is essential, especially when trying to compare protein expression between different samples, to know how much sample has been loaded, as this may not be apparent from the blot alone. For example, when assessing a blot, the band from one sample may appear twice as bright as another sample. This could mean that there is twice as much of the target protein in that sample, or it could mean that more sample or a more concentrated sample has been loaded in one lane than the other. Running a duplicate protein gel and developing the Kumasi stain can help to improve this uncertainty as it will show the amount of total proteins in each sample lane and can reveal any loading inconsistencies. Detecting expression of a ubiquitous protein that should be even between all the samples, such as actin in whole cell and cytoplasmic samples, can also be used as a loading control and helps to ensure consistent transfer of protein samples to the membrane. However, this type of control can be problematic when comparing models in which control proteins are differentially expressed, such as degeneration models. Next, protein transfer or blotting. Proteins must be transferred from the protein gel to an appropriate membrane, typically nitrocellulose or polyvinyl difluoride (PVDF) to facilitate antibody probing. A number of techniques can be used for transfer, including capillary transfer, diffusion transfer, and vacuum blotting, but by far the most common due to its speed and efficiency is electroblotting, also called electroelution or electrophoretic transfer. Here, the protein gel is sandwiched against the transfer membrane and an electric current is applied. Proteins from the gel are carried across and attached to the membrane tightly. Within electroblotting, there are multiple strategies for transfer known as wet, semi-dry, and dry transfer. Wet transfer is efficient and offers flexible buffering, but it is time-consuming. Semi-dry transfer is quicker, but still offers flexibility, but is less efficient than wet transfer for large proteins. Dry transfer is efficient and quick, but offers less flexibility than other methods. Transfer efficiency can be assessed prior to probing using a removable stain such as Ponshu S. Western blot blocking. Due to high affinity of blotting membranes for proteins, after transfer it is important to block any remaining binding sites to prevent subsequent non-specific binding of assay detection antibodies. This is achieved by incubating the membrane with a proteinaceous liquid such as milk or serum. Western blot washing. Following blocking, it is important to wash the membrane between each step to remove excess or unbound reagents. Insufficient or uneven washing can lead to poor quality, patchy blots and high background. However, overwashing can diminish the target signal, so it is important to optimize the number and duration of wash steps. Ensure that the membrane is well covered with an appropriate buffer and apply gentle agitation to wash the membrane evenly without damaging it. Commonly used buffers include tris buffered saline (TBS) and phosphate buffer saline (PBS), often with the inclusion of twin 20 (TBST) and PBST. After washing, the membrane is treated with the primary antibodies. The membrane is treated with the antibody against the target proteins. Only the proteins to be studied specifically bind the primary antibody to form an antigen-antibody complex. After the primary antibody is washed and removed, only the portion of the target protein binds to the primary antibody. The primary antibody treated membranes are then treated with a labeled secondary antibody that binds to the primary antibody and forms an antigen-antibody complex that can indicate the location of the primary antibody and thus the location of the protein antigen being studied. This is followed by detection and visualization. Now let us look into some more details of Western blot detection. While it is possible to use direct detection, that is, a single antibody that recognizes the target and is detectable for a Western blot, more often indirect method is employed. Here, a primary antibody is used to probe a membrane and bind any target protein present. Then, a secondary antibody is used that binds the primary antibody and is detectable. As with all steps, optimization, in this case, choosing the right antibody and determining the optimal concentration is key to a good blot. When an indirect assay is used, a secondary antibody will need to be applied after washing excess unbound primary antibody off the membrane. The secondary antibody should be specific to the species of the primary antibody. Example, mouse anti-rabbit if the primary antibody was derived from a rabbit and possessed the necessary conjugate for chosen detection method. There are multiple methods for detection and subsequent visualization of Western blots including colorimetric, chemiluminescent, fluorescent, and radioactive detection. Both colorimetric and chemiluminescent detection require conjugation of an enzyme to the detection antibody and are considered very sensitive techniques. Horseradish peroxidase and alkaline phosphatase are commonly used enzymes with horseradish peroxidase generally favored due to its stability and amenability to most conjugations and also its low cost. During detection, a substrate is added to the membrane which is acted upon by the conjugated enzyme, bringing about a chemical change. If performing colorimetric detections, a chromogenic substrate is chosen that will produce a change that can be visualized and imaged directly. However, prompt imaging of the blot is important as colors will fade as the blot dries. In chemiluminescent detection, the signal produced only lasts as long as the reaction between the enzyme and substrate is occurring, typically 1 to 24 hours. During this time, the signal can be recorded by exposing X-ray film or by using digital imaging to make a permanent record. In fluorescent detection, the antibody is conjugated with a fluorophore rather than an enzyme. When light of a specific wavelength is shone upon them, they become excited and emit light of different specific wavelength. This can be captured using digital images such as avalanche photodiode APD, photomultiplier tube PMT, or charged coupled device CCD camera. While specialist equipment is required to undertake the excitation and detection steps, there is no substrate step in fluorescent detection, shortening the protocol. It is also possible to multiplex fluorophores within a Western blot assay. Radioactive detection, where a radioisotope is conjugated to the detection antibody and emitted radiation is detected on X-ray film, was extensively used in the past. However, the technique requires special handling to protect personnel from the radiation, is expensive, and has limited shelf life due to radioactive decay. Therefore, the technique has mostly been replaced in favor of other available detection methods. If quantification is required, digital imaging with CCD camera-based devices, for example, offers a better option due to their larger dynamic range, greater sensitivity, and higher resolution. Exposure times can be fine-tuned without the need for repeated time-consuming X-ray film exposures or developments optimizing the signal-to-noise ratio. Now let us look into the application of Western blot in HIV diagnostics. The Western blot has traditionally been the most widely used method for confirmation of HIV diagnosis with a sensitivity and specificity over 99%. In Western blot, the viral proteins are separated by polyacrylamide gel electrophoresis and transferred by blotting onto a nitrocellulose strip. The strips are then reacted with the test serum to determine if any viral proteins are recognized by patient antibodies. This shows a typical Western blot positive for HIV-1 with different reactive proteins. Serological differentiation between HIV-1 and HIV strain 2 can be done using detection of specific antibodies against the HIV strain 2 transmembrane protein GP36. Western blot assays are reported as positive, negative, or indeterminate. A positive result means any two of the P24, GP P24, that is the core protein, GP41, that is the transmembrane protein, or GP120 or 160, the precursor protein and the external glycoprotein bands are present. A negative result means there are no bands on the blot. An indeterminate result is anything in between, commonly an isolated P24 band. If a Western blot is reported as indeterminate, then additional testing such as a P24 antigen ELISA or an approved HIV-1 RNA assay like the gene probe Aptima viral load assay should be considered to confirm HIV infection. Advantages of Western blot. It is a very sensitive test. It gives nice, clean, and easily interpretable output. There are thousands of commercially available antibodies which makes the performance of Western blot easy. It even tests denatured proteins, so changes in functional tests, that is, receptor phosphorylation, acetylation, methylation, etc., can also be detected. Disadvantages of Western blot. The method is not very quick and is slightly labor and intensive. Tissues must be homogenized to release the proteins before performing a Western blot. These are a few of references for further reading. Now, let us discuss some MCQs on Western blot. Western blot tests are the primary method for diagnosing HIV infection. A. True. B. False. Answer is B. False. What is the main limitation of ELISA tests in early stages of HIV infection? A. They are too expensive. B. They are painful to perform. C. The body may not have produced enough antibodies yet. D. They are not specific to HIV. Answer is the body may not have produced enough antibodies yet. Which of the following is the primary method used by ELISA tests for HIV diagnosis? A. Detecting HIV particles directly. B. Detecting HIV genetic material RNA. C. Detecting antibodies produced by body in response to HIV. Or D. Measuring the number of white blood cells. Answer is C. Detecting the antibodies produced by body in response to HIV. Thank you for listening.